All right, so just to give a general description, the final exam review, we're going to be covering mostly the urinary, the endocrine, and the reproductive system. So in the morning, we'll be doing the reproductive, and then most likely we'll be doing these in the night session. Okay, and both will be recorded, so if you can't attend the night session, I will have it posted by the next day. So to go over the logistics, the lecture exam is comprehensive, which means it looks like this. It's going to have 100 total questions. And what you're going to see is the first 50 questions are all multiple choice. Okay, and that's more of the comprehensive portion of it. In other words, everything that was on exam 1, 2, and 3. And it's all multiple choice. The questions are still all looped together. So the first 10, the first set of questions are going to be on this chapter, next chapter, so on. So because we do cover so many chapters through exam 1, 2, and 3, there's going to be about 3 to 5 questions per chapter. So you can see it really is just a wide overview of what we've covered already. Okay. So I would not stress as much on the old stuff as I would on the new stuff. Okay, because each of those chapters only make up 3 to 5 questions. Do review the old chapters because you still want those 50 points, but don't spend the majority of your time on that. What you should spend the majority of your time on is the other portion, which is also 50 points, of which 40 are multiple choice and 10 are fill in the blank. So think of it as an exam 4. Okay, and these 50 questions are going to come from the urinary system, the endocrine system, and the reproductive system. So you can see that of those 50 questions, there's going to be about 13 questions, 13 to 15, whatever makes up 50 per chapter. So this is going to be a lot more detailed. Okay. So I would focus, if I were a student, I would focus the bulk of my studying this week on this content.
So I have a question.
Yep.
So don't concentrate, I mean, kind of know about the central system, but not focus on it. Is that what you're telling me?
I would say that you study however you would like, but if I were a student, this is what I would do, because the bulk 50 questions is on these 3 chapters. So by studying these 3 chapters, I have a better chance at the 50 points, and then study the old stuff, because I should already know the old stuff. Hopefully you're getting that sense of like, oh, it's just a review, because things have been building up on each other. There might be topics that you don't understand quite yet, like the nerve impulse conduction. But the point is you only have a week left to study. So if I were doing that, I'd be like, okay, I'm just going to forfeit those 1 to 2 points and focus on these 10 points. You see what I'm saying?
Okay, thank you.
Yeah, but again, you study whatever method you would. I'm just sharing what I would do if I were a student.
Okay.
You study whatever, because I don't want you to come back and say, oh, Kim said not to study this, and I didn't, and now I'm failing the class. No, that's not what I'm saying. I'm saying if I were a student, this is what I would do. I'm not telling you as an instructor what you should do. An instructor would say study everything.
Okay.
A student would say, let's go with the best strategy. Okay. So my Professor Vu hat would say study everything. But my student hat says, I only have a week, I can't study everything. So where would be my best bang? Okay. Other questions? Other questions on that? Then for the lab exam, this is also comprehensive. Okay. There's going to be 50 questions. About 25 are going to be on the old stuff. Oh. And about 25, it might be off by like 1 or 2, will be on the new stuff. And the old stuff is pretty much everything that was on the lab exam 1, 2, and 3. And the new stuff is on the urinary and repro, because we didn't have a lab for endo. Okay, so you would probably expect 10 for urinary, 10 for female, and 10 for about, you know, up to 25, 10 for male reproductive. Okay, any questions about the lecture or the lab exam? So same thing here. Okay. Then I'm really stressing this because I noticed that there are still some students who are not doing the environmental scan correctly. They're kind of just having it scanned, them staying in one place. And particularly for the final exam, I have to really enforce if you don't have a proper environmental scan, I cannot grade your exam, and you cannot remake it, you cannot take it over again. Okay, so let me give you an example of what an environmental scan would look like. I can't do that because oh, maybe I can. Let me get out of this. Stop sharing. I'm going to turn my camera on. And I need to change my background. Or do I just make my regular background? There you go. Okay. So this is my office. So I would scan my desk so that you can see that there's nothing there except for that. And remember that you cannot have a cell phone. So this right here would not be okay. There should be nothing on your desk except for a clear desk and a blank. If you have a piece of paper, it should be blank. And then I would show that's what's important is I want to see your surroundings. Okay, so there's nothing on either side, because sometimes a student would just show me one side and not both sides, and that, you know, can be very suspicious, especially if I have a red flag for your exam. Okay. Questions about that?
Yes, I only have 6 seconds, or I don't know if anybody else, but I only get 6 seconds on the scan. So I don't know if it's me that you're asking, but I try my best to scan to the left and the right and up and down within 6 seconds.
Yep, look. 1, 2, 3, 4, 5, 6. It doesn't take that long. Okay.
Okay. Thank you.
Yeah. All right. So there's that. Let me share my screen again. Okay. Now I need to see my face. So let's
I have a quick question about the scan.
Yes, ma'am.
Mine typically lags. Like, let's say I'll move and then it like freezes for a minute, for a couple of seconds, and then it starts showing. That's fine, right? As long as I was able to go all areas.
Yes, exactly. I just want to see your surrounding, that there's nothing near you. No cell phone, no paper, no books, you know, just a clear area. Yeah. And then I've been alerted that sometimes, here, let me stop this again. Stop sharing. Okay. And I've also been alerted that sometimes, start video, students may have like their cell phone on their lap. So maybe, you know, like that. And then they'll look down and like this and use their cell phone like this, you know? So just remove any possible doubt out of my mind.
So really, as long as we're keeping ourselves like focused on that screen or maybe looking down to write on our paper, it shouldn't red flag us, right?
Yes, exactly. Exactly. Like if you're like this, there's no red flags. Okay. But sometimes I see students going like this and they didn't scan, particularly that area that they're looking at. That's a red flag. Okay. All right. And again, like I said, I don't ever look at your videos unless there's a red flag. Okay. I
One
don't
other
have time for
question.
that. Go ahead.
Are we allowed to still have piece of paper and a pen to show it in the scan, correct, for the lab or even for the lecture? Can we have it for the lecture too?
You can have it for both, and you can have as many pieces of paper but for every piece of paper that you have to hold it up and scan and show that it's blank on both sides.
Okay, thank you.
Yeah. Yeah, I would have a piece of paper when I'm taking a test because I just need to jot things down before I forget. Share. And then I meant to share screen. Okay, so because this is the final exam, I'm gonna have to really, be very strict about that policy, that request. Okay, so for both of those, you want to make sure that you have the respondents locked down. And webcam with proper environmental scan. Okay, and take my video off because I have to put my face real close to the camera. It's right on the computer. Okay, any questions? Any other questions about that? Okay, so again, really stressing that because I can't grade your exam unless all of that is met. Okay. All right, so I'm gonna go straight to the reproductive. We'll start with the female reproductive system first because it's the most difficult, and then we'll move on to the male. Okay. So for the female reproductive system, make sure you understand the following terms. Gametes refers to the sex cells, and specifically it's the sperm and the egg. Fertilization. What does fertilization mean?
It's when sperm and egg meet.
Yeah. Okay. So when the sperm penetrates into the egg. Okay. What does zygote mean?
The result of fertilization.
Yeah. So this is the new cell that reflects the combination of DNA from the sperm plus the egg. Okay. What does chromosome mean?
Can you explain that to me? Because I don't really know what it means.
Okay. So chromosome is a form of DNA. It's just your genetic information, but it's in a particular form. So DNA just means that it's a, double helix of the 4 basic bases, right? Adenine, thymine, guanine, and cytosine. But chromosome is the form where it's all bundled up, okay? So you can have DNA that looks like this, that just like loose strands, okay? But chromosome is all of those loose strands combined together in a particular order that forms chromosomes. So like a set of DNA together is, let's say, chromosome 1. Another set is chromosome 2. Okay, so it's bundled DNA essentially. Does that help?
Yes, ma'am. Thank you.
Yep. And there are, within the chromosomes, you have 46 chromosomes total, 23 pairs. Are they the same or are they different?
Different.
they're actually the same. So you have 46 total chromosomes, but because you have 2 copies of everything, so here's chromosome 1, here's chromosome 1, here's chromosome 2, here's chromosome 2, here's chromosome 3, here's chromosome 3. So it is appropriate to say that you either have 46 chromosome total or 23 pairs. It means the same thing because they are identical to each other. Does that make sense?
So the 23 pairs are equal to each other. Like each pairs are they're equals, right?
The same. So within the 23 pair, like the first pair is chromosome 1, chromosome 1. The second pair is chromosome 2,
And they're
chromosome
the
2.
same.
Yeah. Okay. Now within those pairs though, that's a great question. It turns out that they could be all identical or they could not. And that's because the 23rd pair specifically is the sex chromosome pair. Okay, so of those 23 pairs, pair 1, pair 2, pair 3, pair 4, so on, up to pair 22 are what we call autosomal. So pairs 1 to 22 are autosomal because they are identical to each other. Chromosome 1, chromosome 1, chromosome 2, chromosome 2, chromosome 3, 3, so on. The 23rd pair contains the sex chromosomes. They may be identical. For example, they contain X and X, or they cannot be identical, in which they contain what?
X and Y.
Exactly. Okay, but you still have 23 pairs. It's just pair 1 to 22 is autosomal, and the 23rd pair is called the sex chromosome pair because they can be identical, X and X, or they cannot be X and Y. What does X and X tell about you?
That it's female,
Yep. And XY?
male.
Male.
Yep. So we don't go into super a lot of details, but sometimes because of the copying error, you can have XXX, XXY. Lots of different issues could happen within the, but this is just showing you like a general what happens on average. Okay. All right. Then we move on to knowing the 2 forms of cell division. There's mitosis and meiosis. So I had a question on that, so I'm going to focus on that a little bit more. I'm going to add a slide. I'm going to do a new slide. Okay, so if we were to look at this, mitosis and meiosis. Okay. Oh, meiosis. I forgot the E. Okay. So both of these are forms of cell division. So how are they different?
Meiosis happens with sex cells and mitosis happens with all other cells.
Yes. Type of cell is one difference. Mitosis is all cells except gametes. And meiosis only occurs in gametes. So let me ask you, what's the difference between all cells versus gametes?
The number of chromosomes.
Yeah, yep. Okay, what's another difference? Come on, we got some smart people. Yeah,
Mitosis,
go ahead.
mitosis copies and divides, and meiosis divides, or at least meiosis 1 just divides.
There you go. Okay. So in terms of the steps, meiosis is copy and divide. Did I say meiosis or mitosis? Divides. So mitosis is copy and divide. In meiosis, you have meiosis 1. It has 2 stages, and meiosis 2. Okay. So in meiosis 1, it just divides. In meiosis 2, it copies and divides. Okay, so here you have 1. In mitosis, you have 1 copy and divide. In meiosis, you have 2 steps. First, it divides, followed by a copy and divide. Okay, so what's the result then?
Mitosis results in a diploid cell, and meiosis a haploid cell.
Sure, yep, because it divides here first, and then it copies and divides. You're absolutely right. So it's going to result in how many cells?
Oh,
But you're right though, how many cells, and the fact that these cells are diploid, meaning they have 46 total chromosomes, where in meiosis, how many cells result? And it results in haploid cells that are 23 total chromosomes, not pairs, okay? Total chromosomes. Can someone help me fill in the blank?
Was it 2 and 4?
Yeah. Absolutely. Now, are these 2 cells identical or not identical?
Yes, they're identical.
Yeah, so these copies are identical. These 4 copies, are they identical or not identical?
it depends.
No,
I
never
I think they
depends.
are identical, right?
They're
They're
not
identical.
going to be identical.
Oh, yeah, okay.
Okay. So, and that's good because meiosis happens in gametes, which means you're going to produce like new progeny. So you don't want identical progenies. Well, in some organisms, humans, we don't. Okay, and that's why there's not one identical person. Even if you're identical twins, there's still some differences that develop. Okay. Questions? Where the purpose of mitosis is just to build within that one organism. Okay. Or since we'll say about humans specifically, when you get to fungi, when you get to bacteria, it's a little different, but in humans, mitosis is to build that one human, meiosis is to build children, progeny. Okay. All right. Does that help you understand mitosis and meiosis a little bit better?
Can you go over just like a little bit in plain terms? How is it that they combine to form the zygote? Like as far as the cells, which cells
Yeah.
go into?
Yep. So that one I'm going to go through in what we call oogenesis, and then what happens there. Absolutely. So I'll apply it to the female and male reproductive systems. But for now, do you understand the basics of mitosis and meiosis?
I think so.
Okay. All right. So then we're going to go through the functions of the female reproductive system. Make sure you know the organs. We're not going to cover this in great detail, but whatever is in your notes is fair game. So be able to describe the ovary, the uterine tube, the uterus, vagina, and other external female organs. This is the one that we're going to go through in detail, oogenesis. Okay. Now, oogenesis is divided into actually there's three, but there's two main ones, ovarian and menstrual, and that's what we're going to cover. Okay. All right, so just the basic anatomy. What would these be right here?
The ovaries.
Yeah. And if you see this on your lab exam, because there's a pair of them, you need to indicate what?
Which side? So right or
That's right.
left?
Exactly. Okay, so
Okay.
don't forget that on a lab, if there's a pair of the organs, you need to identify whether it's right or left. Okay. Then you have this. What is number 2 in general?
The uterine tube.
I love that you use that term. It used to be called the
Fallopian.
yes.
Fallopian.
Oh, I'm switching. Okay. Let me. So Dr. Fallopian is who got the credit for identifying it, and we don't call that anymore by people's names. We call it by what makes more sense, which is why we go by uterine tube more than fallopian tube. What's A, 3A? Oh, oops, what did I do wrong? What is 2A, 2B, and 2C? It looks like it's connected, but it's actually not connected. There's space in between there. So that's why you need this portion right here to sweep the egg once it's released into the uterine tube. What do we call A?
The
Very
fem-
good.
Fimbriae, thank you. And B is just kind of what connects it. And it actually has a little, like a little swelling right here that's not shown. That kind of, that looks like that. So it should be more like this. So what's B? I'm sorry, I didn't catch that. What was B?
the ampulla.
Perfect. And then C is just kind of like an island that connects the rest of the uterine tube to the ovary. What would you call that?
Must.
Yep.
Okay.
There's a T in it. Isthmus. Yeah. Okay. And then this is number 3. I should use a different color. 3A, 3B, 3C. What's 3?
The uterus.
Mm-hmm. A.
A is the fundus.
You notice we use that term pretty often, right? So anytime you see that little curve, it's the fundus. Okay. B.
The body.
Yeah. And
And
C.
C is the cervix.
Yeah. Okay. Then you have 4. What's 4?
The vagina.
Yep. Okay. And now we're just going through this very quickly. Okay. Make sure you know like the tissue layers and anything specific to that organ. All right. Let's go through oogenesis. So oogenesis is the development of the ovum. Okay, and here you see the basic steps here. You see mitosis, you see meiosis 1, meiosis 2, and first meiosis, another way of saying that is just meiosis 1. Second meiosis is meiosis 2. Okay, so you start out with an oogonia. What's an oogonia? How would you describe an oogonia?
Would it be like a zygote or the gamete?
It's actually neither. So this is like the prehistoric cell, okay? So this is the cell that is it starts all the cells that will eventually become a gamete, okay? Because it's so flexible, it's the stem cell. Okay. Now, in the female reproductive system, it's very, very interesting. It's one of the coolest processes in the human body. This starts when a female is being formed in her mother. So this actually starts during fetal development. Okay, so during fetal development, this process is already starting. So as a female is being developed in her mother's womb, she is developing the oogonias, and then the oogonias is actually starting to differentiate. And the word differentiate means to like change. Like the best analogy I can give you about that is, you know, when there's a baby, right? The baby can be anything, and then as the baby ages into like a toddler, they start to walk, they start to be able to do more things and have more fun, you know, be able to do more things. And then that toddler's can go to school and eventually to high school. And then when they go to high school, they can go, oh, okay, I know now I want to be something in the medical field. And then they become a college student, and the college student then decides, oh, I want to become an OTA, and so on. Okay, so that's differentiating. It's the same cell, but it's picking up specific functions until it becomes exactly the cell it wants to be. Okay, so this is like a baby cell, the oogonia. When it's a baby cell, the oogonia, the stem cell, it can be anything, okay? But it decides, it changes, and as it changes from an oogonia to a primary oocyte, that's like a toddler, to the secondary oocyte, that's like a high school student, and then to an ovum, that's like a college student, okay? So it's the same cell that we're following through, and as we follow through, the same cell changes and then picks up more specific functions as it differentiates, as it, you know, ages. Does that make sense? Okay, so the oogonia cell is a form of stem cell. Oops. It's a form of the stem cell that starts to develop during the female's fetal development. So as the female herself is developing in her mother's womb, she's making these eggs, what we call the oogonia. That's even a precursor of the egg. It's a stem cell. And then these stem cells are going to copy and divide to produce more. And she will actually make all the pre-egg cells she will have in her lifetime, all during her own fetal development. How cool is that? Questions?
I just had a quick question. In our notes, so is stem cell and germ cell used interchangeably?
Yes.
Okay. Thank you.
Yeah. It just means a cell that can still divide. It's flexible.
Okay.
Yep.
Thank you.
Yeah. Do you know what the most flexible stem cell? And this is the issue of a lot of ethics. It's a fetal stem cell. So that's huge ethics, and because fetal stem cell, we right now as adults, there's a debate as to whether we still have any stem cells or not, and it's showing that we do have stem cells, but they're not as flexible. the most flexible stem cells, in other words, really it can be any cell that exists, is the fetal stem cell. And that's why there's a lot of controversy and debate about whether like some families like have children that have certain conditions that they're born with, that they oftentimes, that not oftentimes because it's not ethical, but the debate is, should they have another baby so that they can use that baby's stem cell to fix their other child. But very interesting stuff. Look up stem cell replacement. Okay. All right. So anyways, back to this. So during fetal development, and that's the woman, during her own fetal development, she's going to make all the oogonias that she will have in her lifetime before she's even ever born. Okay. So to make more oogonias. So the oogonia right now, it is diploid. Remind me what diploid means again? What does diploid mean?
There's 46 chromosomes.
Yeah. So 46 chromosomes total. Or what we call 23 pairs. And because there's 23 pairs, we use the term 2N. So chromosome 1, chromosome 2, 3, 4, so on. That's the N. And the fact that you have 2 of them, that's where the 2N comes from. So an oogonia is diploid. And to show that, we use the term 2N. Okay. So it currently has 2 copies of everything. Okay. All right. So the oogonia is going to undergo mitosis, which essentially is what again? What happens in mitosis?
Copy and divide.
Yes, ma'am. So it's going to copy and divide. Okay. So temporarily that 2N, the oogonia, is going to copy. So when it copies, it becomes 4N. Okay. And then when it copies, so that now that's copied, it's going to divide. And when it divides, it's 2N. These 2 cells right here are identical. And that is mitosis. So let me ask you a question. So after mitosis, do I have 1 cell, 2 cell, or 3 cells?
Is it 3?
Ah, that's why I asked, because I wanted that answer. It's actually 2.
Okay.
Because the original cell copied, and then the original cell divided. So there's no more original cell.
Okay.
It becomes these 2 cells. Okay. So you completely nailed that misconception, that most common misconception, that it results in 2. And I'm sorry, that it results in 3. Because you have the original one and then you have the 2 new ones, right? No, the original one divided itself. So it no longer is, there. It becomes 2 cells. But then these 2 cells are identical. And whatever genetic information you had in this original cell, you have in these cells as well. But now it just made a copy of itself. Like you had one birth certificate and you went to a copier and you copied another. They're identical to each other. Okay? Does that make sense?
Yes.
Okay. Now, one of these cells goes on to become this cell, what we call the primary oocyte, because remember, it differentiates. Remind me what differentiates mean again? differentiate. It becomes different, right? And then becomes different by, it's the same cell, but it picks up a specific function. Like a baby becomes a toddler becomes a high school kid, it's the same cell, but it picks up more function. So this cell right here and this cell, so this cell right here and this cell, they're the same cell, but now the difference between this cell and this cell is that it's now called the primary oocyte because I know that it gets even closer to becoming an egg cell. Okay, where this other cell right here goes back to this state, which means this cell right here will do what? Will continue to do what?
Copy and divide.
Yes, you got it. Okay, so when there's two cells that are made from an oogonia, one goes back to being an oogonia and continues to copy and divide until it makes as many eggs as that female will be born with. Where the other cell is going to start going towards, you know, changing a little bit so that it will eventually become an egg. Questions on that? Okay, so that's why we say from an oogonia, under mitosis, you're going to end up with what we call a primary oocyte. That primary oocyte is going to start meiosis 1, but not complete it. Okay. So what stage of this female's life have we talked about so far?
Only fetal development.
That is right. Okay, so anything above that line is still fetal development of that female who's producing these eggs, not of the egg itself, okay? So the primary oocyte during the fetal development does start meiosis 1. Oops. Starts meiosis 1, but it does not complete. And that's because at this point, the female is born. So once the female is born, this whole process stops. What starts this process again? So you still, so once the female is born, she technically is born with the primary oocyte because it has not completed meiosis 1 yet. Aye. So when she's born, technically she's born with the primary oocyte because it has not completed meiosis 1. It started, but not completes it. So what helps it to complete? Puberty. Yes, exactly. So at puberty, I should make that sign a little bit lower. What's happening at puberty? If you have children, you'll know at puberty they start to change, right? What causes that change?
Hormones.
Yeah, exactly, hormones. And what hormone in particular is responsible for this?
Estrogen?
Nope. Not at this stage. But keep that in mind though for later.
So FSH?
Yes. What does FSH stand for?
Follicle stimulating hormone?
That's right. Follicle stimulating hormone. It's a stimulating, right? Now we've only focused on the egg so far, but in the next stage, we're going to focus on what surrounds the egg, which is follicles. Okay, so FSH is what drives at puberty this process to continue each month. Okay, so FSH is going to cause the primary oocyte to complete meiosis 1 and become a secondary oocyte. So after meiosis one, from a primary oocyte, it's going to become a secondary oocyte. Now look at that. I want you to focus on this specifically. From a primary oocyte to a secondary oocyte, tell me what you notice in this graph.
They change to haploids.
Yeah. Yep. So you have one cell, one primary oocyte that became how many secondary oocytes?
One.
Technically, there's two. Okay, because if you take 2n and you divide it, you end up with n, right? So technically, there's two right here. There's one right here, and there's one right here. Okay, so essentially what I did in meiosis one, I just whoop, divide that cell in half. So when I divide that cell in half, I will have one cell that's 1n and one cell that's 1n. That's meiosis one. Let's go back to our definition. Meiosis one, what happens in meiosis one? All it does is divides.
Right.
Yep, and that's what we see here. It just divides. So meiosis one is just divide. That cell just divides and splits in half and becomes two cells that are 1n each. Okay? Yes or no? Does that make sense or does it not make sense?
So once it divides, now they're called secondary oocytes.
Yes, ma'am. Okay, is that acceptable? Does that make sense? Okay, now I'm going to add a little bit more to it. Okay, so have you heard the term quality over quantity? Yeah, it's a pretty common American term,
Yes.
quality over quantity.
Yes.
Okay, so I'm willing to bet you that was based on oogenesis and spermagenesis. Okay, because in sperm it's about quantity, in egg it's about quality, because whatever that egg is going to be, that is the house that will become the new however it is able to progress. Okay, so technically in this drawing, these two cells right here, these two secondary oocytes that form look the same, right? It actually doesn't. Okay, so I'm going to make a little box right here and show you what the secondary oocyte actually looks like. That's what it actually looks like. Okay, so meiosis is a way to get rid of one of the chromosome sets. Okay, all the organelles, all the cytosome, all of the good stuff stays in one cell, and the other cell is just a way to get rid of that extra chromosome. So only one actually is the secondary oocyte, where the other one becomes what we call a polar body. And it's the polar body that disintegrates, or what we call undergoes atresia. In other words, disintegration. So you really only have how many cells that move on to meiosis 2?
Just one.
Just one. Questions?
So there was a question on the quiz this week that was about that. It said, what was I'm trying to think of the actual real question, but it did talk about how it eliminated, what was the process of elimination of the chromosomes?
Yep.
And what was it, the polar body?
Yes, ma'am.
Okay.
Exactly it. Now it makes sense, right?
Yes. Seeing that little tiny cell makes it a lot easier to understand
Yes. Yeah,
that it's quality over quantity.
that's right. Okay. Is everyone comfortable with meiosis 1 before I move on to meiosis 2?
Yes.
Yes.
Okay. So the secondary oocyte is also what is released. Okay. So the secondary oocyte is what is released from the ovary in a process called,
Ovulation.
yes. So how many, typically how many cells, how many secondary oocyte is released per month in ovulation?
One.
One.
And that's why, because the other one becomes a polar body. Okay. All right. So then this actually never happens. Okay. So now, once it's released, this is the form of the egg as a secondary oocyte that is released from the ovary to the uterine tube, to the uterus, and if nothing happens, it will be released through menstruation, like out of the woman's body. It will be, leave the woman's body as the secondary oocyte. The only time it will change after ovulation, okay, the only way that will make that secondary oocyte become an ovum is what?
Fertilization.
That's right. Okay. So in order for that secondary oocyte, which is what is released in ovulation, goes through the uterine tube, uterus, okay? Now, when it's moving through the uterine tube, okay, if it gets fertilized, it will then undergo meiosis 2, only if fertilization happens. If fertilization happens, we don't have to talk anymore about this because it's just, it leaves as a secondary oocyte and it's expelled out of the body through menstruation as a secondary oocyte. If fertilization does happen, boom. Okay, so if fertilization does happen, then it will continue with meiosis 2. Okay, now you tell me, what is meiosis 2? What happens in meiosis 2?
You get rid of one chromosome?
Yes, but there's something that happens before that.
Copy and divide.
Yes. Okay, so let's kind of go to the side right here. Excuse me. All right, so I have this one end right here, the secondary oocyte, right? The egg penetrates it, okay? But the combination of chromosome between the egg and the sperm has not happened yet. It cannot happen until that secondary oocyte undergoes meiosis 2. But it does need to be fertilized. In other words, the egg has to penetrate it in order to stimulate meiosis 2 to happen. Okay, so we're not even looking at the chromosome in the egg. I'm gonna make it, that's too, let's see, let's redo that. Okay, So the chromosome of the sperm is the red end. The chromosome of the secondary oocyte is the black end, okay? So I'm not even gonna look at the combination yet because it cannot combine until the secondary oocyte undergoes meiosis II. It just needs that that sperm to penetrate it in order for that process to be triggered. But let's focus on just that secondary oocyte, okay? So we said that it undergoes meiosis II, which is a copy, right? So temporarily, let me make a different color. So temporarily, that secondary oocyte will have how many chromosomes if it's a copy first? You have one end, you make a copy of it, now it has how many?
46.
Yeah. So you can say that it has 46, or you can say it has 2N, right? It's the same thing. All right. So that's copy. And then it's going to divide. Okay. Then once it divides, one of that second side of chromosome right there, that one end is going to become a polar body. Leaving it with this one end to combine with this end. Okay. So this is technically the ovum. The results after meiosis II, and immediately after that, it's going to become 2N, but the 2N looks like this. And that's what we call a zygote. Okay, so one more time. After meiosis I, you end up with a secondary oocyte. Technically, 2 cells are made, but one becomes the polar body. The secondary oocyte is released through ovulation. If it's not fertilized, it continues on as a secondary oocyte and is expelled through menstruation. If it is fertilized, that entry of that sperm triggers the secondary oocyte to undergo meiosis II. Okay, ignoring that the chromosome from the sperm, just that one end of the secondary oocyte doubles, becomes 2N, splits, ejects one pair of that chromosome, becomes a polar body, leaving one with the full cell. And then it's going to combine the chromosome from that ovum, and it's going to combine the chromosome from that sperm and become a zygote. Questions?
So this is where they get 23 chromosomes from each, right? 23 from the male end and 23 from the female end.
Yes, ma'am. And that's why it goes back to being 46 total chromosomes, half from mom, half from dad. And it's this version, this new combination of these chromosomes that will then continue on a whole bunch of mitosis, becomes the brain, becomes the eye, becomes the heart, becomes, you know, who that that human organism will become. It's all because of the combination of these two sets of chromosomes. Questions? How about what doesn't make sense?
I wouldn't say that it doesn't make sense, but what is a little confusing to me is, I guess in meiosis two, when it is one end and then it turns into two end, when it, I guess, is fertilized.
Yeah, okay. Let me make a new slide for that because I'm running out of room on this one. All right. I think that was Kendra, and that was an excellent point. So you have, just let me stick to the color right. Okay, so all gametes have to go through the complete meiosis, because you're probably wondering, like, meiosis two, there's no point to that. Why can't it just, it's already at one end. The sperm is already at one end. Why can't it just combine? That's just not how it works. You know, sometimes you have to go through that process because that is, you know, programmed into the cell. So technically, you didn't have to go through meiosis two. You could just use that one end in that state from the female and the end from the sperm, right? And combine it, and boom, you'll be done, right? But that's not what the cell is programmed to do. So that's why it has to undergo meiosis two, or meiosis, whatever way you want to pronounce it. Okay, because technically you're probably, I did too. I'm like, oh, it's already at one end. Why not? You know, why go through all these extra steps? Because it's just programmed that way. Because gametes are programmed that way. And it makes more sense when you get to the spermatogenesis portion of it, because it goes through the exact same meiosis one, meiosis two. That's just what gametes are programmed to do. But anyways, okay. So one N is the equivalent of 23. Not pairs, 23 total chromosome. Chromosome 1, 2, 3, 4, just that one single. Because remember, after meiosis one, we divided it, so we don't have that anymore. Okay, so we have 23 total chromosomes. In meiosis two, it's got to go through two steps. The first step is a copy. The second step is a divide. But the requirement for it to undergo meiosis two is that the sperm penetrates the egg. Okay, so here's that sperm. It penetrates the egg. Okay, so when that sperm penetrates that secondary oocyte, it's going to trigger the secondary oocyte to undergo meiosis two. What happens first in meiosis two? It's going to copy. So from 1N, it's going to become 2N. In other words, from 23 chromosomes, it's going to become 46 chromosomes. Does that make sense? So that's the copy. Okay. So here's still the sperm. Okay. Then it has to divide. So from this cell, from the secondary oocyte, it's going to split. Back to being 1N or 23 chromosomes on that side. And this one, same. This is the polar body. It doesn't have any of the good stuff that a cell needs to maintain itself. It doesn't have cytoplasm. It doesn't have organelles, nothing. Just a way to get rid of that chromosome because it copied. Okay. Here you have, here you have the ovum. The ovum is the completion of meiosis 2. Goes back to being 1N and 23. Why? Because that's what the cell is programmed to do. At this point, okay, the ovary plus the sperm that has penetrated into the egg will combine its genetic information. And when it combines its genetic information, magic happens. It is now 2N, resulting from an N from the sperm and N from the ovum. And that cell we call a zygote. Does that help?
Yes, ma'am.
Okay. Whew, okay, that's hard stuff. Okay. So, okay, but we gotta move on. We only have like a few minutes left, so
Can I ask just one quick question?
yeah, go ahead.
Just to make sure, like when we say, okay, females ovulate once a month, really we dispose of a secondary oocyte, right?
That's
If
correct.
it's not fertilized, it's not necessarily, okay.
Yep, that's absolutely right. Other questions? Okay. All right. Then I'm going to add, please do ask all the questions you have though, because I need to add on another layer of difficulty on this same thing. So I need you to be 100% comfortable with this before we move on to the next part, or at least 90%. Okay. Then to add on to that, because it's not difficult enough already, we have to look at follicular genesis. Okay. So follicular genesis follows the development of the follicles which surrounds the egg. Okay. So during fetal development, okay, so this is fetal development, so before birth right here. So before birth, we learned about the oogonia, right? We learned that the oogonia undergoes mitosis, produces 2 cells. One becomes a primary oocyte, the other one, excuse me, the other one repeats the cycle, goes back to being an oogonia and undergoing mitosis until that female has enough to sustain her for her whole lifetime. Okay. What we didn't talk about is that surrounding that oogonia are follicles. Surrounding that oogonia are follicles, is one follicle, and that one follicle is made up of follicular cells. So the whole structure is a follicle that's made up of all of these follicular cells right here. Do you see that? If you were to describe this epithelium, what would you describe this epithelium as? Is it simple or is it stratified?
Simple.
Okay. Is it columnar? Is it squamous or is it cuboidal?
Squamous?
Squamous would look something like this.
So it's cuboidal.
Yeah. Yeah. Oh, oh, you're right. It's supposed to be squamous. I drew it wrong. Let me undo that. Maybe I can erase it. Should draw it. All right. There you go. It's too small. There you go. So, there. So you are right. It is simple. Ay. Squamous. It does not become cuboidal until after. Okay. So the follicular cells at this point are simple squamous. Okay. All right. So that is also the type of follicle that surrounds the primary oocyte. There we go. All right. So, excuse me. So where you see the change in the oogonia becoming a primary oocyte, the follicle that surrounds the oogonia, called the primordial follicle, doesn't change as the oogonia changes. Does that make sense? So the oogonia undergoes mitosis and changes and becomes a primary oocyte, where the primordial follicle just stays as a primordial follicle that surrounds the primary oocyte. Questions? Okay, so the primordial follicle stays as a primordial follicle during fetal development. Then at birth, it stops and it continues at puberty or adolescent. So at puberty, that primary oocyte finishes meiosis 1 and becomes a secondary oocyte. During this time, you see a lot more changes happen to the follicle. Now it makes more sense, right? What hormone is responsible for this?
FSH.
Yeah, follicle stimulating hormone. And what do you have? You have a follicle. So at puberty, when FSH is released, it's really targeting the follicle. And as the follicle changes, that's what drives kind of the continuation change within the oocyte. Okay. So at puberty, the FSH is going to cause the primordial follicle to become a primary follicle. So from primordial, it becomes a primary follicle. So let's look at the picture and see what the differences are. So look at the diagram and describe to me what some of the differences between the primordial follicle to the primary follicles are.
Looks like the simple squamous becomes simple cuboidal.
Yes, that's the mistake I made, right? These pictures are a little bit more difficult to see, but yeah. So definitely, so it transitions in shape from squamous to cuboidal. Okay. And the cells that form that follicle is no longer called the follicular cells. It is now called the granulosa cells. Okay. So that's one of the major differences between the primordial follicle, which transitions into the primary follicles. Now, here's the cool thing, because remember we said that the female's born with all she'll have in her lifetime. So each month, the FSH stimulates only about 20 to 25 of these primary follicles. And of course, if it's the primary follicles, it also includes the oocytes. So only about 20 to 25 of that primordial follicle / primary oocyte undergoes this process. Okay, and it really is a race to see which is the healthiest, most robust combination of follicle / oocyte that will actually be released. So truly, in a female reproductive cycle, it is completely about quality. Okay, so at the beginning of the cycle each month, the FSH is going to stimulate about 20 to 25 of these combination because the follicle surrounds the egg, okay, to go through this process. So at the beginning, FSH is going to cause the primordial follicle to become primary follicle, and the main difference is changing in shape. The primary follicle is going to develop further into a secondary follicle. Now look at the diagram. Can you tell me what the differences are between this image and the second image?
Simple becomes stratified.
Yeah, exactly. So it becomes layered. And there are additional structures that develop, which include the zona pellucida and the thelica folliculi. It's a little bit more detailed than I had you know in the notes, but it's good to know what these things are. Okay? Just to know that these are some of the differences that also form as it develops from a primary follicle to a secondary follicle. Okay, but the key difference is that it becomes stratified. That's what you'll be tested on. Okay. What is important to know is that what will lead into the next part is this zona pellucida right here. The zona pellucida is this cell matrix, right, or this like substance right here, this gel-like substance in between. Okay, so that zona pellucida is going to become this structure right here, which is the antrum. Okay. So when you look to see what the difference between a secondary follicle to a tertiary follicle is the formation of that antrum. That's the key difference. So here, between the primordial to the primary, squamous to cuboidal. From primary follicle to secondary, simple to stratified. From secondary follicle to tertiary follicle, the antrum. Okay.
Is another name for the tertiary follicle mature follicle?
Technically, no. So it does go through the stages of primary, secondary, tertiary, and then mature follicle right here. But in our book, in our notes, I simplified it and just eliminated the tertiary because there's not much difference between the tertiary and the mature. The main difference between the tertiary and mature is the one of the 20 to 25 that reaches the final stage. Okay, so think about this way. So you have 20 to 25 of these structures developing, and whichever one reaches the mature stage first is the one that will eventually be the finalist. Okay, because here's what happened. We haven't gotten to the hormone portion of this content yet. So what happens is that the follicle stimulating hormone is going to continue to cause the development of those 20 to 25. But within those 20 to 25, one of them is going to become mature first. So you'll have 20 to 25 tertiary follicles, but one of them will become mature. And when it becomes mature, it's going to stop FSH. which means all the other 20 to 25 that haven't reached mature state will not reach mature state. So it's a race. Whichever one matures first, in other words, it doesn't need FSH anymore, is what triggers the FSH to turn off. And all the others that are lagging behind it never finishes the development. So at the end, you only have one mature follicle. Does that make sense?
Yes, ma'am.
Okay, so that's why the tertiary follicle really, there isn't a difference, a major difference between the tertiary and the mature. It's just that of those tertiary, one becomes mature, and that's the one that we focus on in our lecture. So from the secondary, we go into mature. Okay, so of that tertiary, only one of them finishes first, and that's why we call it the mature follicle. So from the secondary follicle, you have one mature follicle. Okay, and that mature follicle, because that antrum, that fluid space builds up pressure, it's going to cause that follicle to rupture and releases the structure, releases the oocyte, the secondary oocyte. The follicle stays back, and the follicle becomes the corpus luteum. So the mature follicle becomes the corpus luteum. Okay. Now, once that secondary oocyte is ovulated, we don't have to follow the progression of the follicle anymore, because now it's just the oocyte. And that's when we follow whether it is fertilized or not. So then really there's a split right here between what happens to the egg and what happens to the follicle, because now it's separate entities. Okay, we know what happens to the egg, depending whether it's fertilized or not fertilized. We need to finish what happens to the follicle. So what is the follicle now called?
The corpus luteum.
Beautiful. We have to follow it because it has a function. What does the corpus luteum do? Yeah, what does the corpus luteum do?
It secretes the progesterone.
Yeah. And why is progesterone important?
It helps the lining of the uterus
Yeah.
build up.
Look at that timing. This is why this is one of the most beautiful processes in the human body. Why would you want to line the uterus? Oh, I should finish writing uterus one.
For a fertilized egg, if it gets fertilized.
Exactly. So it knows it just expelled the egg. Now it's preparing each month. It doesn't know whether it will be fertilized or not, but it's preparing in case it happens. Each month, that lining becomes thick, filled with blood to nourish the potential zygote that may form. If it doesn't form, that's cool. It will shed through menstruation. If it forms, it's got a really nice house full of nutrients ready for it. Okay. At this point, where's your FSH level? High or low?
Low.
Yeah, exactly. Okay. So FSH is high in the beginning. It's going to continuously go down, and it's going to go down and stop once the follicle becomes mature. What takes over then? Instead of FSH, what takes over?
Progesterone.
Not quite yet. Luteinizing hormone, LH. Okay, So that mature follicle is going to be stimulated by LH, which causes it to rupture and release the egg through ovulation. So LH becomes high. And then once the egg is ovulated, LH will start to go down, and LH is going to be replaced by progesterone. Okay, so I want to show you this image right here. This is a beautiful image. Okay, no, seriously, it includes kind of two main things we've talked about so far. So all we've talked about so far is the ovarian cycle. Ooh, that was a whole bunch of stuff, right? And all within the ovarian cycle. So we talked about the follicle, and we've talked about the oocyte. We have not gotten to the uterine cycle yet. Okay. So all we've talked so far is the ovarian cycle. I started to introduce hormones, FSH, which you can see is dominant in the first half right here. Followed by LH, which is dominant in this particular part. Okay, and then it is replaced by progesterone levels and estrogen levels. Okay, and we have not gotten to the uterine yet. So does everyone follow what we've talked about so far? Questions before I move on to the uterine cycle? Does this picture make more sense now, though, that we've gone through all of that?
Yes, ma'am.
Okay. Yeah. So when I was going through this class, the same thing. If I looked at this picture initially, I'd be like, oh, okay. And then I talked about, oh, I'm like, oh, that's what those things mean. That's the feeling you should be experiencing. If not, ask me questions. You should understand why FSH is having those arrows to the primordial follicles, to the primary follicles, to the secondary follicle. You should understand why LH is targeting the mature follicle, ovulation, the corpus luteum. And then you should be understanding how the corpus luteum starts to have arrows to progesterone and a little bit of estrogen. And then you should understand why the progesterone is targeting the lining of the uterus and causing it to become thicker. Okay. Questions? So essentially in the ovarian cycle, you have two main parts. You have the follicular phase, followed by the luteal phase. Right? Where in the uterine cycle, there's 4 phases. You have the menstrual phase, the pre-ovulatory phase, the ovulation phase, and the post-ovulatory phase. And then there's the menstruation, proliferative phase, the secretory phase, and the menstruation. We'll go through all of them. Okay, any questions before I continue though? Okay. The best way to kind of go through this is if you look at in here, there is in the supplemental materials, I actually created this graph Supplemental reproductive. Let's go in here. Female right here. Here we go. This, here we go. This is what I made for my students. And it is a really nice kind of visualization of the oogenesis, what happens to the egg, the follicular genesis, and the uterine wall, what happens to the uterine wall based on their cycles and how they are paired to each other. So this is a really nice chart to kind of, have you seen this before? Has anyone accessed this before?
Yes, ma'am.
I did.
Awesome. This really is this is your key study guide here for the female reproductive system. So you can see that along the uterine wall, during the follicular phase, during what's happening to the egg and to the follicle, this is what's paired to that. So you can see that while the egg is forming and the follicle is developing, you have the menstrual phase. So the menstrual phase is when the stratum functionalis sheds. Okay, from day 1 to day 5. And you do need to know these days. Okay, so there will be a test question. So like, for example, the menstrual phase during day 1 and 5, what happens there? Okay. Following that, day 6 to day 13 is the proliferative phase. So now it's preparing again to support. So now it's preparing. I don't know why it keeps erasing. And now it's preparing again to support the lining, to support the new secondary oocyte / potential fertilized egg that will come. So here you can see that the term estradiol right here is just another word for estrogen. So estrogen is going to cause the regeneration of the stratum functionalis. Okay, nothing happens in ovulation. Okay, then during the secretory phase, that's when the egg is already released. You have the corpus luteum, which starts to produce progesterone. So progesterone is what causes that lining to thicken. And then if pregnancy happens, then the luteinizing hormone, which causes the corpus luteum to stay about, because the corpus luteum is going to decrease. But if pregnancy does happen, the HCG, the human cortical gonadotropic hormone, is going to replace the LH, and that's what keeps the progesterone being released. And as long as the progesterone is being released, the stratum functionalis stays along for 9 months. So for about 9 months. Questions? So the uterine lining is a little bit more simple. Okay. So that's what happens here. So day 1 to day 5, menstruation. Day 6 to day 11, 12, you have proliferative phase. And then you have the secretory phase, which we talked about in terms of the progesterone causing the thickening of the lining. And then you have the post-ovulatory phase. Okay, and that's it. Questions on the female reproductive cycle? Do you have a few minutes to stay and do the practice quiz? Or does everyone have to go?
I can stay.
I can also stay.
Okay.
I can see also.
All right.
I would love to stay, but I have to go to work.
Okay. Well, if you can come to the evening session, if not, that will be recorded as well. Okay. So for the urinary system, you want to make sure that you're able to listen and describe the functions of the kidney, which include filtering blood. And my pen's out of battery. All right. Let me use my mouse. Okay, so it includes filtering blood. And when it filters blood, it means two things. One, it's going to clean it, and then it's also going to remove the waste. So all the blood goes into the kidneys. Okay. And remind me what components of the blood is there?
I stepped away to get a pen. Could you repeat your question?
Sure. So we're just going through the functions of the kidneys, and one of the main functions of the kidney is to filter blood. So filtering blood refers to the movement of blood into the kidneys to not only be clean, but also to remove the waste. So I just wanted to review what is in blood. What would
Red
you
blood
say?
cells,
Red blood cells. Okay. What else?
white blood cells,
Mm-hmm.
plasma,
Mm-hmm.
platelets.
Okay. So really you have solid stuff, X, and you have the liquid components. Okay. So within the liquid components, let's look at plasma. What would you find in plasma? So all the water, what else?
I've drawn a complete blank.
That's okay. Others?
Proteins,
Yes, plasma proteins. What else? Nitrogenous waste. Hormones.
electrolytes,
Yes.
glucose,
Glucose. Yeah, you read my mind. Glucose. Amino acids, etc. Okay, so really it's this stuff right here that's moving into the kidneys. Okay. All the other stuff like white blood cells, red blood cells, and platelets, they should stay out. They're not going to be filtered. Anything large shouldn't filter into your blood. I'm sorry, into your kidneys. So if it filters into the glomerular glomerular capsule, that means that it could potentially be found in your urine, right? Because whatever isn't reabsorbed, it moves its way through to becoming urine. Should you ever find blood in your urine?
Not normally.
Not normally, exactly. So you should not see red blood cells specifically in your urine. If you do, that means that there's something wrong, that filtering system is damaged, inflamed, or infected that allows the red blood cell to move through. Okay, so really what you should find that is filtered into your kidneys, or specifically your glomerular capsule, should be just the plasma. Okay, so within all that component, there's good stuff in there, but then there's also waste. So your kidneys, specifically your glomerular capsule and your nephron in general, knows how to be able to separate it, to separate the stuff that you want back into your body and the stuff that wants to continue on and then excreted it out of your body. Does that make sense?
Yes,
So
ma'am.
awesome. So think of it this way. You know, you wore your shirt for all day long. You have, like, you spilled food on it, you sweat into it. So you want to wash your shirt, right? So what do? You throw your shirt into what?
The washing machine.
Exactly. So you throw your shirt into the washing machine and it's going to like go through its cycle, right? And it's going to remove the bad stuff, the dirt and the sweat, right? But should your shirt be intact so that you can take it back and use it again?
Yes.
Exactly. So that's very similar to what your kidneys do. It takes your blood, and in that blood, or specifically in that plasma, there's stuff that you need back and stuff that you want to get rid of. So it separates it. So that's what the cleaning means. The cleaning means it separates the good from the bad, or the stuff that you don't need in your body anymore. And then once it separates it, it brings back the clean stuff, and then it removes the stuff you don't need in your body. Okay, just like a washing machine does. You can take your shirt back, free of sweat and dirt. Okay. All right, so that's what filtering blood means. There's two parts to it. There's just, because a lot of times when people think of filtering blood, they only think of like the cleaning, but not the removing of the waste. So it does both. Okay. So in addition to that, it also regulates water volume. And by regulating water volume, I don't know why I'm not able to write anymore. What is it doing?
Affecting blood pressure.
Yes. What else? I'm just going to put blood pressure so we don't forget. Blood, BP, blood pressure. Because if you have too much water in your blood, what happens to your blood pressure? Up or down?
It rises.
Yes. So if you have too much water in blood, it rises. If you don't have enough water in blood, in other words, you urinate all of it out, what's going to happen to your blood pressure?
It's going to go down.
It's going to go down. Exactly. Now, water is really interesting. Whenever water is pulled in to the blood vessels or left out, what does it affect? What other things does it pull? Does it move with it?
Is it the electrolytes?
Yeah, it's going to also affect the movement of the electrolytes, okay, as well as other substances, smaller substances like hydrogen ions. And that's why when it's regulating water volumes, it's also going to affect the pH level as well. How much hydrogen is kept in blood, in the plasma. Make sense? Sorry about that noise. My sister's coming in and out of the house. Questions? Okay, good. So then another topic that you'll want to know is the gross anatomy of a kidney. So be able to describe what the fibrous capsule is, what's the hilum, what's the associated blood vessels, the renal cortex, the renal medulla, and the renal pelvis. And actually, if we were in class together, I would actually dissect a kidney for you, and you can see all of those things without using a microscope. So all of those things are the gross anatomy, and it's called gross anatomy is because you can see it, okay? Where microscopic anatomy would be the nephron. So in order to see really the parts of a nephron, the smaller networks, you'll need to have a microscope. So you also want to know what the nephron is. It's considered to be the functional unit of kidney. Why is it called the functional unit of kidney?
Because it filters the blood.
Yeah, it's what does all that work. So when we say functions of the kidney include filtering blood, regulating water volumes, regulating pH, it is the kidney, but more specifically, it's the nephron. So that's why it's called the functional unit, okay? Because it's what does all those functions. So the nephron is really key. So you want to make sure you know all of its parts and the function of those parts, which include the renal corpuscle. So the renal corpuscle technically is part nephron and part of the blood vessels. So the renal corpuscle includes the glomerulus and the glomerular capsule. Which of those is actually a part of the nephron, the first part of a nephron?
The capsule?
Yes, very good. So the glomerulus then is the blood vessels, okay? Then another portion of the nephron is the renal tubules. The renal tubules is just one tube, but depending on what section, it has a different name, okay? It's one continuous tube, but depending on the section, it has different names. And not only depending on the actual area, but those areas are different in what they're lined with. And that's why it's particularly important to know the lining of the parts of the renal tubules, because the structure affects the function, okay? So you'll find the PCT. So following the glomerular capsule, you will find the PCT, or the proximal convolute tubule, then the nephron loop, and then the DCT, or the distal convolute tubule. And then depending on what book you look at, some books consider the collecting duct as part of the nephron, others do not. Your textbook, Salden, does not consider the collecting duct as part of the nephron, okay? But just so that you know, oftentimes, the parts are as follows: the glomerular capsule, the PCT, the nephron loop, the DCT, and the collecting duct, okay? All right, so know those structures and then know the lining, because the lining will affect its function, okay? You should also be able to describe the process of urine formation. Okay, there are 3 to 4, depending on, again, what book you look at. But there's three basic ones. Your textbook adds a 4th, though. So the 3 basic ones would be filtration, reabsorption, and secretion are the 3 basic ones, but your book adds a 4th one. Does anyone remember what the 4th one is?
Water conservation?
Yeah, that's really key, actually, because if you learned about the urine formation process and you left out the water conservation or the water reabsorption portion, it would mean that we would sit on the toilet the whole day, okay? Because we filter a lot of blood, but we only urinate a very small percentage of it. We filter, depending on whether you're male or female, anywhere around 150 to 180 liters a day. That's a lot, okay? So, but what you urinate should only be about 1 to 2 liters a day. So without that water conservation or the water reabsorption, you'd be sitting on the toilet the whole day, okay? All right, so those are the 3 to 4 steps of urine formation that you'll want to be able to cover. And then describe the structures outside of the kidney, which include the ureter, the urinary bladder, and the urethra. And same thing for this. You want to make sure that that you go through the structure, the function, and the layers, and then describe the steps in micturition process. So in other words, the urine, the peeing part of it. Okay. Any questions before we move on? Okay. So the gross anatomy, I'm going to skip just to kind of save us time and go right into the nephron. Okay. So when we're looking at the nephron, we have the following parts. Okay. So we have A, B, C, D, and E. Can someone tell me what A is?
Globule, globular capsule.
Perfect.
Okay.
The glomerular capsule. Okay, so we're looking at parts of a nephron. And A being the glomerular capsule. Okay. Now let's spend some time on the glomerular capsule. Okay, so the glomerular capsule is lined with what? What's the glomerular capsule lined with? Let's start with this, simple or stratified?
Simple.
Very good. Cuboidal, squamous, or columnar?
Cuboidal.
Yes. Okay, so it's lined with simple cuboidal epithelium. And in particular, the inside of that glomerular, think of the glomerular capsule like a baseball mitt. I don't know if that's how you draw a baseball mitt, but anyways, think of it like a baseball mitt, okay? So there's the inside and then the outside layer, the parietal and the visceral, okay? So the inside layer of the glomerular capsule is made of a very particular cell. It looks something like this. And then you have another cell here. Actually really pretty. Okay, so the simple cuboidal epithelium that you find on the inside of the glomerular capsule, these things right here, that looks like squid or aliens right here, they're referred to as what? You should know this term. Yay, podocytes. Excellent. The podocytes have these extensions called dendrites, and then together they kind of form like this. So do you see this area right here? In between those dendrites, do you see the area I'm lining in red? Yeah?
Yes, ma'am.
Okay. So those areas are called filtration slits. They help to make sure that only the fluid component of plasma move through and into the glomerular capsule. Okay, so the filtration slits are formed by the podocytes. The podocytes are the cells that make up the simple cuboidal epithelium. Let that sink in. The podocytes, which form the filtration slits, make up the simple cuboidal epithelium. When I talk about podocytes, I'm talking about the cell level. When I'm talking about simple cuboidal epithelium, I'm talking about the tissue layer. Okay, so they can be known as, they can have two different names because they're not really the same thing, right? So the podocytes are the cells that form the tissue. And that's why when you talk about the lining, whether you're talking about the respiratory lining or the urinary lining, there's a difference between the name of the tissue versus the name of the cell. Okay, so let's apply that concept. So this morning, I saw a few of you joined this morning as well. So this morning, I talked about the follicle, okay? So the follicle is formed by a tissue, and then that tissue is formed by the cell. So if we're talking about the primordial follicle, the primordial follicle is formed by what tissue? This is the one I made a mistake on, if you remember.
It was simple squamous.
Exactly. What are the cells that form that simple squamous epithelium?
Is it granulosa?
It does become the granulosa once it becomes the primary follicle, but the primordial follicle consists of follicular cells. Okay? So kind of keep that concept, because students, that's one of the big concepts that students get confused. They're like, podocyte, simple cuboidal epithelium, is that two different names? No, it's not. We're not referring to the same thing. It's different levels. One, you have the tissue layer level, and the other one's the cellular level. Okay? All right. That was kind of worth it. 3 minutes of my time. to kind of go through, because it's going to be consistent. That concept is going to be consistent through all the chapters. Okay, so pay attention to what I'm asking for on exam question. Am I asking for you to name the tissue, or am I asking for you to name the cell? Okay, all right. So glomerular capsules, the first part of the nephron, it consists of simple cuboidal epithelium tissue made up of podocytes. Okay, and why is that important? Is because the podocytes have these they form the filtration slits that help to only allow small things like liquids to pass through. That's why normally you shouldn't find red blood cells or white blood cells or platelets or even really large proteins in urine, because it shouldn't be able to make it through these filtration slits. Okay, all right. So that should get us good for the glomerular capsule. What's B?
The PCT.
Yep. Or the proximal convoluted tubule. Okay, so the proximal convoluted tubule is proximal, and even if the picture looks weird, like it's upside down, left to right, right to left, whenever you find the glomerular capsule, immediately whatever follows it is always going to be the PCT. So you just follow it along. Okay, follow that tube. Okay, so even if like this portion right here overlaps this portion, as long as you follow the tube, because it only goes in one direction, you will always be able to distinguish what's the PCT from the DCT. Okay, all right, so PCT, what's important about this?
It has the brush border.
Yep, what else?
It's where the majority of the absorption occurs.
There you go. Majority of reabsorption occurs here. Reabsorption happens along the whole tubule, from B to C to D to E. But the majority of reabsorption happens in the PCT. Why is that if it's the same tube? Oh, there we go. Why is this?
Is it because of the brush border?
Yes, partly. What else?
This is the closest to the glomerular.
It's
Closest to
not.
the
Good
capsule?
guess. That's a very good guess, but that's not why. So let's go back to the brush border. What do you mean by brush border?
Is it the microvilli?
That is it. It's the lining. Okay. The lining differs depending on whether you're looking at B, C, D, or E. Okay. Remember what I said, structure informs function. And that's why you have to learn about the lining, because it makes sense. The majority of reabsorption function happens here because of the lining structure. So what is it about the lining of the PCT that you don't see in the other portions of the tubule? So first of all, what is the lining that you would find in the PCT?
Simple cuboidal.
Yep. Yes, ma'am. Well, what's unique about it?
The microvilli.
Yeah. Have you heard of microvilli before?
Yes.
Yeah. Where did you hear about microvilli before?
In the small intestine.
Yes, exactly. So in the small intestine, what did the microvilli allow it to do?
Absorb
Yes,
nutrients.
exactly. Absorption. You're going to save me so much time. Absorption. Absorption, exactly, because you find in the digestive system, right? So that's the first time it moves into your body. Okay, so the first time that nutrients and stuff is put into your body, that's through the digestive system, and that's why we call it absorption. This right here in the urinary system, it's the second time we're putting it back into the body. which is why we call this process what? Not absorption, but
Reabsorption.
that is it. So in A, glomerular capsule, you have filtration. In B, C, D, and E, you have reabsorption. But the majority of the reabsorption occurs in B. Why? Because that set of tissue has microvilli, where the others have less to none. Isn't that cool? How structure informs function? Okay, all right. So simple cuboidal epithelium with microvilli, hence the majority of things are reabsorbed here. Okay, all right. Oh, okay. Let's see, the lining, major. Okay, that's good. Oh no, we should finish our thought. So we know that the majority of reabsorption happens here. Okay, and what kind of things are reabsorbed? So you want to make sure that you know. In your book, there's this really cool page, and I will get you the number to that page. You know, I'll just get you the image for that. There's an image that shows you what exactly is reabsorbed, secreted, and filtered. Reabsorbed, secreted, and filtered. Do I have it in here? I usually have it in here. Ah, here we go. Okay, so so here you can see at the PCT, this is happening, reabsorption is happening, and a little bit of secretion is happening. But overall, the movement, whether it's moving in or out, occurs at the PCT. Why? You can tell me now, why, right? Why?
The microvilli.
Yes, ma'am. Lining. We're at the next part that we're going to go to next, right here. See? Only these things are moving back and forth. You can see a lot less, right? Which tells you that what's going to happen to the lining. Is it going to stay the same or is it going to change?
It'll have less microvilli or no microvilli.
Perfect. Yep. And what else? Look how thin this area is. It's still going to be simple, but now it's going to be lined with what?
Squamous.
Yeah. Okay. All right. So let's go back here. All right. So make sure you look at that that image on slide 6. Okay. That will show you what exactly is being moved back and forth. Okay. All right. Then now for things to be able to move back and forth like this, okay, the reabsorption and the secretion right here that happens in the PCT, what kind of transport mechanism is it going to use? So now we're not looking at why, but we're looking at how. How are things moved?
With ATP?
Yes. It's active transport. And if it's active transport, it's going to require ATP. Very good. Okay. C. What is C?
The nephron loop.
Yes, ma'am. Okay. And the nephron loop has 2 parts. A. Oh, let's not do letters. Let's do numbers. 1 and 2. What's 1?
The descending limb.
Make sure you put descending limb if it's on the lab exam. Okay, some of you just put the descending loop. It's got to have this descending limb of the nephron loop to be safe. Okay, so descending limb of the nephron loop. And then 2.
The ascending limb of the nephron loop.
Mm-hmm. Yep. Okay. You can see that in both the descending and ascending, they have the thick segments, and then they have the thin segments. Okay. So here's the thick segment. Here's the thin segment. Okay. So in the thick segment, what do you think it's going to be lined with? Simple or stratified? Mm-hmm. Shape?
Cuboidal.
Yes, ma'am. So at this point, is there going to be any different with the microvilli? Is it going to be the same, less?
It's gonna have less.
Yeah. And you can see that a lot of it is actually the thin segment. So the thin segment is going to be consistent of what?
Simple squamous.
Mm-hmm. Okay. All right. So here you have mostly movements of water and electrolytes. Okay. So water, electrolytes, and a little bit of urea. But notice that the urea is a secretion more than a reabsorption. Okay. But we'll add it because it is something that's being moved. Okay. So with water, how is water moving? How's the water moving? How's the water being reabsorbed?
Osmosis.
Yes. And we know that osmosis is what? Active or passive?
Passive.
Requires ATP or no ATP?
No ATP.
Perfect. Okay. Now, when water moves, it causes this pulling effect. Okay. So when water moves, water through osmosis, so water moves through osmosis, but when the water moves, it's going to pull what? Pull some of the salts with it, particularly what? Sodium. And when the sodium moves, it's going to pull what with it?
Fluoride.
Yeah. Okay. Because remember, things like to move from high to low concentrations. Okay. So when the water moves, it's going to pull sodium. When sodium moves, it's going to pull chloride. Okay. Questions? All right. How about D? I'm going to have to move to this side.
D is the distal convoluted tubule.
Yep. DCT. And I love that you said it, the full name. Still convoluted tubule. What's acceptable on the lab exam?
It has to be spelled all the way out.
Yes, very good. Just a reminder, because I saw some of your quizzes, you did not spell it out. Okay, so you have to spell it all the way out. And what is this lined with? Let's look at its function. Only this much is reabsorbed, which tells you what? It's still thicker, but only this much is reabsorbed. So look at function and then guess, if you don't know, what the structure is made of. Huh?
Simple T-Bortle.
Yep. Tell me more. With microvilli or without microvilli?
No,
Very
no.
excellent. No microvilli. That explains why not a lot is being reabsorbed. You still have some. Okay, some absorption does occur without without needing the microvilli, but less. Okay. And then what's E?
The
The
collecting duct?
collecting duct.
Yep. And it would be the same. Okay. Okay, questions? All right, you got it. Now I'm going to add one more to this. Control copy. Control paste. This. There's a fast way of doing this. Oops. That. There you go. Okay. So you have the nephron. We labeled, we identified, and then we went through details. And now I'm going to add the blood vessels to it. Okay. Questions before I move on to the blood vessels? Okay. So on the inside, because the glomerular capsule is like a mitt, right? Like if you throw a mitt, someone catches the ball in a mitt, the mitt surrounds that ball, right? So the ball in this case is the glomerulus, okay? But before it became the glomerulus, it starts out like this. A much larger blood vessel. When it exits the glomerulus, it wraps itself extensively around the renal tubules. Okay. Okay. 1, 2, 3, 4, 5, 6. Can you identify one for me? What goes into the kidney? What blood vessel enters into the kidney?
The renal artery.
Yes. Renal artery. And it transitions. And if you took the advanced A&P, I'd have you memorize what it transitions to. But for the sake of this class, I want you to jump directly from 1 to 2. So what's 2?
The
What's this?
afferent arterial.
Yes, exactly. Okay, you study blood vessel. You know it goes from artery to the smallest one, which is an arterial. Okay, how I remember this is the A stands for arriving. That's the one that's before it becomes number 3. Because the A for afferent means A for arriving. What's number 3 then? That's surrounded by the glomerular capsule.
The glomerulus.
Excellent. And the glomerulus is technically a network of what?
Capillaries.
Yes. It shouldn't be memorization. It should make sense because substance is moving from the glomerulus into the glomerular capsule. Okay. And anytime you have movement of substances back and forth, it's always going to be, if it's a blood vessel, it's always going to be capillaries because capillaries are what allows for exchange, for the movement of things. Okay. So if I were to ask you, okay, what is the filtration membrane consistent of? What would you say? So here I have the renal corpuscle, which consists of the glomerulus and glomerular capsule. It forms, the filtration membrane. Help me list it out. 3 parts.
The capillary.
Yep. And the glomerulus is very specialized. The glomerulus is made up of capillaries, but the lining of the capillary is normally what? Capillaries are always lined with what?
The fenestrated endothelium.
The endothelium. You recall that, right? The endothelium. Now, what makes these capillaries particularly special is that they have little holes in them. And the German word for window is fenestra, thus fenestrated endothelium. And this is unique to the glomerulus. Okay, so endothelium is what you'll find in all capillaries, but these capillaries are special or unique because they have little holes in them. So they look like this. Okay. All right, so you got the first lining. What's the second one? What forms the filtration membrane? Just like the respiratory membrane, there's always something in between the two. So the basement membrane. We just talked about number 3, so I'd like you to answer that.
The filtration slits from the podocytes.
Beautiful. You got it. That's what makes up the filtration membrane, and that is on the test in the past, and will most likely be on the test now. But it's randomized, so I don't know if you'll get the question or not, but it's on the test. Okay. All right. So that is number 3. It's a network of capillaries, the fenestrated endothelium. And then that forms the filtration membrane that consists of the fenestrated endothelium of the capillary, the basement membrane, and the podocyte filtration slits. Okay. Number 4, please.
The efferent arteriole.
Very good. At this point, you're going to be like, what? When I was in this class, I was like, what? Because normally, what's the transition of the blood vessels? It goes from artery to capillary to?
Venule.
Yes. That is the normal, that's what I've learned in blood vessels. It should always go from artery to capillary to veins. But this is one of the 2 exceptions in your body. There's only 2 exceptions to that rule, and this is one of them. The other one is the hepatic one, okay? So in this one, this is the exception where it goes from artery to capillary back to being arteries, okay? Or arterial to be specific, okay? All right, so E, how I remember this is E is the one that leaves. It comes after the glomerulus because E is for exiting. Okay, sharing you all my memory tricks, okay? So E in efferent arteriole is the exiting. So hopefully you will always be able to label them correctly because you just find whichever one goes in, whichever one goes out. So the efferent arteriole is always the one that wraps itself around the renal tubules, okay? All right, what's 5? It wraps itself. Yes. Exactly. I should be quiet because you know this stuff. The paratubular. Finish that for me.
Capillaries.
Why is it capillaries? Capillaries always equate to what?
Sites of exchange.
Yeah. Okay. You have the capillary, which is the endothelium. Then you have the renal tubule, which is usually simple cuboidal, some with microvilli, some without, but you have a movement of things back and forth that allows for reabsorption and secretion. So it's got to be capillary there. Otherwise, it's too thick and things can't move back and forth. Make sense? Okay. So it's always going to be the paratubular capillaries, and that's what allows for reabsorption and secretion. Otherwise, where would the substance go? Right? So, and it's called peri because it's going around, tubular because it's going around the tubes of the renal tubules. Okay, so peritubular because it's going around the renal tubules, capillaries because you have to have a blood vessel that allows for exchange. See, again, it's not about memorization. It makes sense if you recognize the structure and the function. Okay. All right, so the paratubular capillaries, that's important to know because capillaries allow for exchange. And that's why you expect reabsorption and secretion to occur here. Okay. And then what's 6? At this point, it's going to leave the kidney. And go back to the heart.
Is it the renal vein?
Yes, ma'am. It eventually becomes the renal vein. Questions? Okay. You have 3 fluids. Well, you have 1 fluid, but goes by 3 different names. What are the fluids called?
Plasma?
Yes. 2 more.
Glomerular filtrate?
Yeah, filtrate. And I like that you added glomerular, but I'm going to call it filtrate in general here. And I'll talk about the glomerular filtrate in a little bit though. Filtrate. And one more.
Urine?
Yeah. It's the same fluid. Honest to goodness, it's essentially the same fluid. It is the same fluid, not essentially, it is the same fluid. Okay, it may not have all the components, but it's the same fluid. If it's plasma, it's always going to be located in where?
The blood vessels, the
Yes.
capillaries.
Yes, we'll say blood vessels to be generic because, you know, we have capillaries, we have arteries, we have veins, right? So there's so many of them. So we're just going to say blood vessels. Anytime it's in a blood vessel, we call it plasma. Anytime it's where do we call it filtrate?
In the tubules?
Yes, in the nephron. Anytime it's in the nephron, we call it filtrate. And when do we call it urine?
When it reaches the collecting duct.
Yes, collecting duct and onward. This is a test question. What do you call the fluid that's been reabsorbed? What would you call the fluid that has been reabsorbed?
Plasma.
Yes. What would you call the fluid that has been, that goes into the glomerular capsule?
Filtrate.
Yep. So those are the types of questions that will ask you on your knowledge of the fluids, where they are, and what it's called. Pretty straightforward though, right? As long as you remember this. Plasma, always in the blood vessel. Filtrate, always in the nephron. Urine is not until it reaches the CD and onward. Okay, questions? All right. That, having gone through all that, that should make this one much more simpler. So here you see a combination of the blood vessel and the nephron. So now we can talk about the urine formation. Okay. So you can see here that urine formation, this image right here, I like this image more than the one in Saladin, but you can see that this textbook uses 3 steps, where Saladin adds a 4th. Very important, okay? But they kind of lump tubular reabsorption with the water conservation, that's in this textbook, okay? So the first step happens here. What's the first step?
Filtration.
Yes. And more specifically, because it's filtration occurring between the glomerulus and the glomerular capsule, it's called glomerular filtration.
I just have a really hard time saying that word for some reason.
Oh, I did too. I'll be honest. I struggle too, but you know what? It doesn't matter as long as you can spell it. Okay. Honestly, because I've had professors, depending on where I took the class, in the north, in the south, in the east, and in the west, they all have different pronunciations for it. Like respiratory is referred to as respiratory in the south. It's like tomato, you know. So I honestly, you know, there's certain things that I'm very particular about, like calyx, okay, the minor and major calyx. Okay, sometimes people say it different. I wouldn't say wrong, but differently outside of the normal acceptable saying for it. Then I get really annoyed. But you know, certain things are just there's just multiple ways of pronouncing it. But how I do it is that when I'm studying this, I go like this: glomerular. I make these little lines to help me, because if you can say it, you can spell it. Okay, like sternocleidomastoids.
Yeah, that was another one.
Huh?
Yeah, that was another one that I could only spell and not say.
Yes, yes, yes. But if you divide the words up like this, that helps you with both pronunciation as well as spelling. But pronunciation is overrated because different countries, even within the United States, is pronounced differently, and then of course in different countries. So don't worry too much about it. Okay, anyways, back to your information. So the first step occurs in the glomerular in the renal corpuscle. Okay, so the renal corpuscle is that area that I shaded in yellow. It consists of two parts, the glomerulus and the glomerular capsule. Okay, so infiltration, what was the fluid before glomerular filtration, and what is the fluid after glomerular filtration? What was it before? What did it become?
It was plasma and it became filtrate.
I love how you said that so fast. Amazing. That's what you'll need to know for the exam as well. Okay. In the past, that has been a question. So you can see that in terms of glomerular filtration, we have what's called glomerular filtrate rate. Make sure you read the content on glomerular filtrate rate. Okay. The glomerular filtrate rate is really important, particularly once you get into your health field, because they use that to determine how healthy someone is, whether they're eligible for kidney transplant or not, or liver transplant. So the glomerular filtrate rate is used very often. Like if you did a simple blood test, they will say on your blood, the results, GFR. So African GFR and non-African GFR. That is completely racist, by the way, because that's based on the stereotype that African Americans are more bulky, so their GFR rate should be different, but that's complete myth. But anyways, you'll see those values on your blood results. Just kind of see how your kidney's working. Okay, so that's what GFR is, and that's where I talked about how you should know what it means, the definition. Okay, you should know what substances are moved. Okay, and within the substances, you should know what can move and what cannot move. What should you not find in the filtrate? We talked about this earlier too.
White blood cells, red blood cells.
Yep, that's right. And anything really large, right? It shouldn't be able to make its way through the fenestrated endothelium, the basement membrane, and the filtration slits. Only fluid should be able to make its way through, and what you find in plasma. Okay. And then you should know, there are a few numbers that you do need to know. I don't usually make students know numbers, but you will want to hear. So it's about 150 to 180 liters that's filtered. Of this, how much becomes urine?
Between 1 and 2 liters.
Yeah. What determines that range? Sex, males and females.
Okay.
Yep. Okay. Who has the larger urinary bladder, by the way? Because, we're at a St. Kate's, a Catholic women's college.
I have no clue.
No, you should have seen this in the anatomy. Males have a larger urinary bladder because in females, the urinary bladder right here is followed by what? Lying right on top of it. Uterus. So in women, you have to make room for the uterus, so the urinary bladder is going to be smaller. Okay, so if you're a female and if anyone ever says, why do you have to go bathroom, especially men, why do you have to go bathroom so often? You say, because my body is made for the future generations. Okay, it's true though. Women have smaller urinary bladders to make room for the uterus. And that's why when you're pregnant, you have to go pee much more often. Okay. Where males, the whole space is about the urinary bladder. Okay. All right. Just a cool fact, not on the test. Okay. Then you have the reabsorption and secretion. So both of these occur where?
And the tubules.
Yes, ma'am. So it occurs between the renal tubules and the peritubular capillary. Where the glomerular filtration occurs within the renal corpuscle, which includes the glomerulus and glomerular capsule. And we talked a lot about it, right? We talked about why things has to move through active transport. We talked about how water moves through osmosis, why the majority of reabsorption occurs in the PCT. So we covered already in the previous sets. Okay, questions? Okay.